And now I humbly present you very valuable scientist, Dr. Şenay Şimşek. Dr. Şimşek, how are you?
Good, good. How are you? Thank
you very much. Actually, it's a good morning for you, right?
Yes, it's about 7 a.m. here.
7 a.m. for you, and thank you very much. It's a really pleasure to have you today in the this young wannabe scientist in front of the young wannabe scientist. Thank you very much for joining us today.
Thank you. Should I proceed with the presentation?
Of course, after that, we will move to the question and answer session.
Okay. Okay, great.
Where's yours? Thank you very much.
Thank you, and good afternoon or good morning wherever you are. So I live in the United States. Currently, it's about 7 a.m. And before, so the title of my presentation is. Okay. Yeah, title of my presentation is having a look at the 360-degree approach for food chemistry. And also, before I start my presentation, I would like to thank Phyllis for inviting me for this wonderful event. A little bit background about myself and how I ended up in the United States. Before I start my talk, maybe we can recommend attendees to mute their microphones. Okay, so I grew up in Turkey and I have BS degree in chemistry and master's on biochemistry, and I was working on my PhD on biochemistry at Gebze Technical University. It was also Turkish Gebze Yüksek Teknoloji Enstitüsü. And I was working on my biochemistry PhD, but I had opportunity to visit United States for summer to Purdue University. And I decided to quit my PhD in Turkey and started a new PhD in the United States in the field of food chemistry. And I completed my PhD in December 2006. And right after that, started as a tenure track assistant professor at North Dakota State University. So since 2007, I have been living in the United States, and I also climbed the steps of promotion. Now I'm a full professor. And usually at the beginning of my presentations, I use this family picture, and I have 4 children. And my team, of course, I will be showing you some research slides, but anything we do is the results of great teamwork. And by the way, this picture was taken a couple of years ago. As you all know, it is impossible to take such picture these days. And at any given time, I have about 25 to 30 researchers in my group, 7 staff members, about 8 to 10 graduate students, time slip employees, visiting scientists. And we actually have a holistic view of food science and how we can conduct research. This is why I have an animal figure in my slide. This will be the bison grown in our region in North Dakota. And this is the mascot of North Dakota State University. So once you come to campus, you will be seeing lots of, if one day anyone comes to campus, you can see bison mascots around when we have the football games, any games that we have. Our bison is walking around with the mascot. Of course, the students are wearing the bison mascots. As being part of the NDSU, North Dakota State University, my main responsibility is research. So I have 85% research appointment and 15% teaching appointment. For research, I focus on food chemistry and cereal chemistry. I also have an adjunct appointment at University of Puerto Rico and at Purdue University. I teach classes on carbohydrate chemistry, food chemistry, and cereal technology. And I have very close working relationship with food industry. I know some of you from Turkey or some of you from other countries in Southeast Asia, even maybe Europe. I compare based on my experience and compared to other countries. I believe one of the advantages in the United States that industry has a very strong ties with universities, not only for me but other universities as well. It's a great opportunity to develop this type of relationship because as a scientist we have actually access real world problems, and we work with them to solve their problems. And as part of my teaching class, again, this will be some of my students teaching in the classroom. We were visiting companies, doing some hands-on experiments. This will be the applied research. But after COVID-19, now I developed some lecture content, and we are using virtual reality technology to teach some classes. And I work with computer scientists to develop this content. Because now we cannot put so many students in the one room, but we still want to give them the hands-on experience. Using the headsets, they can have this opportunity. And another, let's move on to next slide. And this will be my carbohydrate or food chemistry class. Again, after COVID-19, we are also using some virtual reality headsets to teach carbohydrate chemistry classes. And again, I am also interested using other types of technology for teaching and study chemistry. And this will be the augmented reality. I just a very short video about augmented reality and how I utilize this in some of my chemistry classes. So this will be the Zoom screen that students are using. So the camera is here, and this is, and also they can use themselves, but we are using different technologies in our classrooms. And in terms of research, my program, I always take 360-degree approach, because as a faculty member, we are at the center, and then we have interactions with industry or um, collaborators, not only in the university, but also in the nation and globally. We provide service in the scientific community and also the state that we live in. We provide education, and have a research mission, and it aligns well with the vision of department, vision of the college that I am part of it, and the university. So we have to think everything from different angles so that we can develop a very strong collaboration network. So it's so far so good. Can you still hear me? I don't see the attendees on my screen. If
We can hear you hocam.
Okay, great, thank you. So what will be the type of research that I will be doing, and what is my strategy? So my research is focusing on in-depth investigation of more specifically, I mentioned you food chemistry, but more specifically, I have passion for carbohydrate chemistry. And our research goals include production of high quality research, and we want to address we want to address applied problems, but using a very basic approach. So this will be a research in a nutshell. And I thought maybe we might have some young scientists in the group. They may want to know what type of habits will be good if one wants to be a successful researcher at any given university. So first thing is, I always say this to my students: you cannot force yourself to study a field. We have to have passion for what I do. You don't want to suffer in the lab if you don't enjoy just being there. So it's good that if you have a passion for what you do, and self-directed motivation. And this is these days is very important. Play well with others. What it means is we should get along very well with everyone around us. We call it collegiality in the university environment. um, Sometimes not everyone easy to work with as we imagine. um, if we call that also conflict, and if we it's very normal to have conflict in the workplace, but we should maintain our calmness and try to get along with everyone. This is going to be very useful in the long run. Of course, having a good communication skills is very important. And the research is good, but life is not always about research. We also suggest that students should have something in interest, maybe running, painting, playing football, do something that is going to relax you and take your minds off of from time to time from the science. And we also, one of my focuses is this is I usually say that in the classroom also, just like I do now, I have to communicate my science. I need to talk about my science. Science is not complete until it's communicated to the globe. So we publish our research in different platforms. I attend lots of workshops, conferences, meetings. These days, everything is virtual, but once the coronavirus is over, we will get back to our regular meeting schedules. And you have to develop your network, because then you are known in the field and you can actually share their experiences. So I know this is true most of the time for US, but if any given university position, funding is very important, especially like for my position. I have a very heavy research appointment, so we constantly seek for grant opportunities. And we write grants to multiple institutions, not only government, but also different industry. And this will be my specific mission for my program. And this is what I have been doing at NDSU, North Dakota State University, last 15 years, is globally recognized food chemistry program that is strategic and solving tiny problems. Is basic but also applied. I just included some pictures from my labs, because maybe again some of you might be professors and will be very familiar with these instruments. I just have some of them again. This will be my gas chromatography. This will be ultrapressure liquid chromatography, quadruple time of flight mass spectrometry. Again, different types of chromatographic techniques. This is, I will have a more maybe specific picture, but this is we call that automated dietary fiber analyzer. Um, and because I have an interest in fiber and how we can quantify them, and based on the method, and these will be official methods. When we run a method, we need to make sure that it is following an official procedure widely accepted by certain associations. So I mainly work with Institute of Food Technologies and Cereals and Grain Association, and they have official methods. But based on the methods that we use, your fiber values might be different. So I have again a small video about how we use the fiber analyzer. Here you can see that we have samples and liquid samples inside the bags, and we have a little agitation. So they are moving like this. So we actually here, each bag is acting as a stomach system. And whenever you eat something, you are going to have this similar function, similar agitation in your stomach. So here we are actually creating an artificial digestion system, and food samples are here. It's not in this video, but on the this side of the system, we have we have buffers and some solutions, and they will be your stomach juice and enzyme mixes to control the pH. So we just basically try to create that stomach model, and we can see, quantify how much is digested. After the stomach digestion is done, then the samples are moving to next bag. This will be your simulation of small intestine. And these bags are not a normal bags. They have certain properties, um, and they have filters between them to simulate, again, digestive tract. This will be some of the facilities that I use throughout the university. Advanced microscopy techniques, nuclear magnetic resonance spectroscopy, mass spectrometry. So these will be my facilities. But how, and I mentioned that I work on food chemistry mainly, but how we can understand how carbohydrates are one of the food components are the carbohydrates, right? If a food chemist says I work on food chemistry, it's usually proteins, carbohydrates, and lipids. Again, I have a special passion for food carbohydrates and how we can test them. Again, going back to going back to 360-degree approach. Why? Maybe people, when they eat a loaf of bread, or when you are biting a sweet cookie, you never, nobody thinks about why this cookie is soft. We have some cookies or crackers that are crunchy. So when you like maybe sweet ones or salty ones, when you have a bite, you hear this crunchy sound. And everything, all these properties are controlled by carbohydrates found in the food sample. Even and I'm sure everyone has the experience of making rice pudding or regular chocolate pudding at home. When you add water and start heating, but while mixing, you will see that you have increase in viscosity, right? It becomes thick, and then when you put them in the cups and wait until it cools down, it becomes a very firm texture. Pudding, you have to use a spoon. It doesn't drop and flow anymore. Why? Again, all these properties are controlled by the chemical reactions in the food system. This is what I do. This is what I study. And most of the time, again, those food carbohydrates, they are, where do we get? Most of the time, they are not synthetically produced. They are coming from plant sources. Um, and let's talk about starch, for example. We have starch from wheat, corn starch, or potato starch, tapioca starch. They all have different properties. And if based on the source, those differences, they start and they originate from a field. If you have a dry land versus irrigated, those properties are impacted. So this is why I say here the 360-degree approach. What happens in the field, then what happens when we harvest the product and process it. And once you make a cookie or bread or whatever the purpose is, you are using it, fine structure of carbohydrate chemistry is impacting all these properties. Of course, at the end, it's going to have impact on the final product. But then once you consume and eat, it goes into the digestive tract. Again, fine chemistry of carbohydrates are going to impact how they will be digested. And some of them, now everybody is talking about gut microbiome, gut health. And if it is not digested in the stomach system, how it will be fermented in the large intestine. All these details are impacted by, again, chemistry of the food that you are eating. And we always, again, I just use this analogy of chain reaction, that structure is impacting properties. This will be like starch granules, and then those properties impact to function, how it is going to be cooked or prepared by processors, food makers, and how it's going to impact to functionality and quality of the end product. And I mentioned you, I work with carbohydrates, but I also work with mycotoxins. And so we use the holistic approach in food science to solve, again, applied problems using basic approach. So one of my projects, what did we do? And I also, these will be, Dr. Menti will be my collaborator, and this is one of my students working on her PhD. Um, one of the problems, in the wheat cereal grain industry, and sometimes if we have the right conditions in the field, the grain can be infected with mycotoxins. And for this case, the specific mycotoxin is called deoxynivalenol. And this will be the structure of deoxynivalenol. And if we have the disease, and the wheat has high levels of mycotoxins, it could not be used for human consumption. So up to 2 ppm, they could be blended, but after 2 ppm, between 2 to 5 ppm, it could be utilized for animal feeding. But after 5 ppm, it has to be thrown away. So we thought, can we take this trash grain, because if it has high mycotoxins, it's not going to be consumed. Can we take this grain and then try to remove this mycotoxin that is not good for us. By the way, the other name of this mycotoxin, so it's deoxynivalenol, it is also called vomitoxin. Why? Because if you eat a food product that has high levels of this toxin, it is going to cause vomiting. So that is why it is called vomitoxin. And so we were thinking, can we find a way to remove this mycotoxin and try to utilize this grain? One thing with this mycotoxin, it is distributed inside the grain, but it's water soluble. So we try to use a wet milling process. Wet milling is you are grinding sample using water. Because we haven't heard about this strategy to remove this mycotoxin before, so we were not sure how tightly it will be bound into the matrix of the cereal grain. We add water, centrifuge, use of course mixing and centrifuge, and we wanted to quantify this mycotoxin in the precipitate and supernatant. So I want to talk about multiple results for this sample, but once we removed this mycotoxin from wheat fractions, we wanted to analyze, for example, protein, starch, and different molecules using again different techniques, and we were able to actually extract starch and protein, and they did not have DAN. So DAN is the mycotoxin. So why it is important? Because uh, before we use this approach, this grain was trash again because of high mycotoxins. It is not good to be used as food or feed, even feed. But when we use the wet milling technology and remove deoxynivalenol, and we were able to separate proteins and starch, and as in the pure form, those compounds could be utilized in different applications. So this will be evaluating strategy to mycotoxin infected grain. Again, we use chemical methods, chemistry, to save a product that is going to be wasted. Um, so this will be another project that one of my students has worked on. I'm sure you have heard about biodegradable films. So sustainability in today's world is a buzzword. We hear that all the time. And, and one of the concerns is again using plastic packaging or other types of bags. And I was so happy actually when I visited Turkey a couple of years ago now. If you want to use plastic bags, you have to pay for it. Now everybody is carrying their own bag. I'm sure that it's not pleasant that you have to pay, but I am sure now lots of people are using their own bags and we have decreased in the plastic bag usage. This is not that level. This will be something that has a direct relationship with the food product, but we wanted to develop biodegradable films using the byproducts from cereal industry. And so the corn milling is a very commonly used million dollar worth of industry in the United States. Wheat milling, ethanol production. So these will be the three cereal industry that they have byproducts. Byproduct is once you take your product, like in case of corn milling, it will be cornstarch. For wheat milling, it will be refined fine flour. So for ethanol production, once they produce ethanol, they have residues. So we were thinking that can we make films from these byproducts from different cereal industries. And again, this will be an examples of the films that we made, and we checked um, its surface properties. So once you drop a water, is it water soluble? How water is remain intact on the surface? For example, this had more interactions with fiber. Sorry, it has more interactions with water compared to this. So I am not going too deep into the how we did it, how we done it, but it was just a strategy that we took, and I can expand any of the research topics I am talking today and how we did it. But so for some packaging materials, we want to see it, we don't want it to have interactions with water, and sometimes we want it. And then also we investigated topographic properties using atomic force microscopy. Again, in some cases, we want to have very smooth surface, but sometimes the rough surface is okay, and it will have the less visibility, whatever is inside the package. And of course, we claim these will be biodegradable, so we check the biodegradability of these films, and there is an official method for it. And when we are using active soil means that it has naturally occurring soil microorganisms in the soil. We put the we place this biodegradable film. There's again certain protocol that we follow, and we measure that how much is being degraded. And we so far we had the data for 140 days, and as the days progress, we had increase in the biodegradability. So more than 80% was degraded in some cases, but the corn bran originated biodegradable films were 70% biodegraded by 140 days. So, we even made some suggestions that what type of applications they can have for specific applications. If somebody wants to develop a biodegradable film for dry snacks, wheat industry byproduct, wheat bran originated salvo material could be the best. But if you want to have something stretchy and clingy wrapping material, corn bran originated corn milling industry byproduct could be good. And if somebody wants to again develop a packaging material for dry goods, ethanol industry byproduct, we call that byproduct dry distillers grains, that it could be used. So now I'm switching topics. I just wanted to give information on some topics, and again, we can expand those if there is an interest or question. Um, so one of the other topics, and we actually is on modified starch. And these will be again some of the publications. And for this project, I collaborated with a scientist from Mexico, Sweden, and Purdue University. So with starch, I want to talk a little bit about starch. And this will be native starch with no modification. And starch is found in the granular form. So by looking at under the microscope, one can tell this is pea starch versus wheat starch or corn starch, because based on the botanical origin, they are going to have different size. And starch is a polymer of glucose, just glucose. And it's the only polysaccharide that human can digest. It's the only polysaccharide. It's the only big complex molecule that human can digest. It's a field that people devote a whole their life. There are multiple books about starch chemistry. So like think of that as a cultural. You can find starch in cultural foods. So for example, if you go to Africa, and every culture has a main starch source in their diets, if you think about it. So for Turkey, Middle East, or even Europe, and in the United States or Western countries, it's in the form of bread. Bread is consumed almost in every meal. If you go to Asia, Indonesia, Malaysia, other countries, it is in the form of rice. I mean, you are people from Malaysia, and I know there are some maybe from that country. You are eating, or Indonesia, they eat rice in every meal. It's very important. And in Africa, they are using yam or tapioca type of products. So every culture has a starch-based food, and it comes with culture again. You don't notice what we eat it. Why? Because it is the only polysaccharide we can digest. And the chemistry is well studied. So we can actually use some strategies to modify and change the structure to enhance its end-use applications. Because starch is not water soluble and in the cold water before cooking. And and in some products also starch is impacting I remember I told you like half hour ago, starch is impacting the textural properties of food. The best example, I am sure everyone has the experience, is staling of bread. You bake your bread or you buy from store, couple of days later it gets firm. Why? Because of changes in the starch chemistry. Um, so there are ways to actually change that chemistry of the starch and improve its role in the food system. And one of the strategies we were using is modification of starch using octanyl succinate. So this will be main building block of starch. I told you that it is a glucose polymer. So each monosaccharide here is glucose, and every glucose has a hydroxyl group. So with the chemical modification reaction, we are replacing hydroxyl groups with the succinate groups. And once we do that, then it has the side groups as part of the structure. And then molecule becomes partially hydrophobic. And why this is important? Again, if some of you might have chemistry backgrounds, but if something is hydrophobic, it runs away from water. It doesn't want to dissolve. Just like if you mix water and oil in the kitchen, oil is going to remain on the surface. Even though you shake, you just put it, and if it rests for 5 minutes, you will have the phase separation. Why? Because oil is hydrophobic, water is hydrophilic. But in food systems, sometimes we want that blend, and it is called, and we are using some compounds that can facilitate that interaction. And we call that emulsification properties. That emulsification means that a molecule has some properties that is hydrophobic, and some they have properties hydrophilic. With this modification, we are making starch partially hydrophobic also, so that it can actually have good interactions with hydrophobic molecules. Um, Again, this will be some of the study work that we published, and also once we do the modification, we here for example, I say that we replace the hydroxyl group with octanyl succinate, but I have to confirm that. So we use advanced chemical tools to confirm that we were able to change those, and we have done emulsion studies like you see in this picture. So they start to, like here, oil droplets were cluttering around the starch granule. So what that means that we improve the emulsification properties. So this would have some applications in the food industry. For example, I don't know if it is commonly used in some of the countries on this call, but like salad dressing. Salad dressing is when people make salad and we have all the spices and it is poured onto salad. But one of the issues in this industry, you were having mixtures or phase separation. So anytime you are using it, you just have to shake it so well. But with those type of compounds, you can actually maintain that nice mixture that you have all spices. They stay in the air, in the solution. They don't precipitate. This is just one example. So this field you can have lots of applications. Again, in the food industry. Again, these are some examples of how we confirm that we have this substitution. And for food applications, we have limit. So we have to be careful that this is called the degree of substitution. We always monitor how much hydroxyl is replaced by succinate groups. And then also we are using starch. Again, this will be a starch molecule to encapsulate some of the flavors. And they also have different applications. Um, so the starch films, again, we encapsulate phenolic compounds in this case. And those phenolic compounds were extracted from pecan nut shell or hazelnut skin. And we made films similar to this. And in that case, those phenolic compounds, they had antimicrobial properties, and they wanted to use them on the wounds to prevent um, any inflammation. So these will be the films that we generated from modified starch. And we also check the biodegradability properties. Just this is another project. I will go very fast. Whole wheat consumption is one of the topics that it's very well studied here. And whole wheat bread is sometimes people like the rich flavor. It is very preferred, but in some cases, people don't want to eat because it has that bitter taste. You feel it's yeah. I will just stay with the bitter taste. But we were thinking that maybe we can use some oligosaccharides as a microencapsulating agent to mask those undesirable flavors. So those undesirable flavors, we are going to call them host in this animation. And then and cyclodextrins are cyclic oligosaccharides made of glucose. So here they have this donut shape, and here we have multiple glucose units coming together in the round shape, and they are forming a cyclodextrin. And there are three types. And inside that molecule, if we just look at the molecule here, inside is hydrophobic, outside of the molecule is hydrophilic. And if the size of the molecule, the gas molecule is a good fit, that we can actually form these complexes. And how we know if it is fit or not? So we do molecular modeling studies on the computer with collaboration with chemistry department. So we can do some of this, test these interactions, calculating binding energies in the computer environment. But then we also apply them into the real life and use again NMR to confirm that if we have the successful complex formation. So we know which protons are responsible for the complex formation. Again, this will be my host molecule. This will be cyclodextrin. And once I have this successful complex formation, I will see shifts in the NMR spectra, and I can confirm that we have the complex. Why? And this will be the computational studies, and we had great match with the computational studies with the real in lab experiments. And this is still a work that is in progress. So this is the last example, I think, that how food can impact our health. So here in this project, we generated structurally tailored cell wall polysaccharides from wheat. I'm going to call that arabinoxylan hydrolysates. And we wanted to check their impact on inflammation in the small intestine. I'm not going to go so much details, but we had um, 35 to 50 starting material first, and they all had different fine structure. How I define the fine structure is degree of polymerization, degree of substitution, and we wanted to test those in the cell culture systems if they can prevent inflammation. And we studied the fine chemistry again using some of the tools like NMR. I will skip this one. It's lots of information, but gas chromatography, mass spectrometry for linkage analysis, and we tested them in the cell culture models. And we came up the idea. Our results have shown that fine structure of fiber has impact on the inflammatory properties. So this will be structure and biological functionality relationship. So we found that, and we also, I will show you some other slide, but we found that, based on the structure, they are found, I have the specific signaling pathway in the small intestine. Oh, okay. I didn't include that slide. So this will be the summary slide for this study is some compounds with higher terminal xylose versus what is the fine structure. Some of them, they had pro-inflammatory, some of them anti-inflammatory. Which one is better? Well, it depends. If someone has an issue of too much inflammation, like inflammation gets out of control, like some people, they have wounds and it doesn't heal so well, that anti-inflammatory compounds are good. But if somebody has immune deficiency, then pro-inflammatory compounds are good. Also, we check the same. This will be another study. Looking at the time, I think we are okay. So we look, going back to original picture of 360-degree approach. So far, I have been talking about the cell wall polysaccharide, which is called arabinoxylam. And we had some projects, impact of fine chemistry of that compound in health. And in one project, we prepared biodegradable films from that compound. And then in this project, we wanted to study how chemistry of that compound is changing during bread making process, because it impacts the rheological properties. And actually, there is a lot going on with the chemistry of, of these compounds, and we usually use simple animations, and some of them are water extractable and some of them are extractable. So during the bread baking process, we have structural changes that they impact the rheology of the bread, and then the final quality, because some of the molecules are being hydrolyzed and then they are moving into the, like, water unextractable portion becomes extractable. So why, again, we do different types of studies for the even same molecule, how it impacts processing, how it impacts health, how it impacts to make different types of compounds. But the big picture here is the fine structure, what sugars you have, what type of linkage you have, how do you have the substitution pattern. It can impact function, it can impact application. So one of the things in the United States is Food and Health, a National Institute of Health release next 10 years strategic plan. And one of the major point is they want to focus on food, because most of the chronic diseases are related to our diet, like diabetes, high cholesterol, even cancer, that researchers have found strong interactions between the food and how it impacts our lives or our health. So there will be lots of research coming up even in the future, how food can impact health. And it could it be used as medicine? Can food could be used can food be used to treat some of the diseases? We call it precision nutrition. How many times you have heard some people say, I eat anything I want anytime, I don't gain weight. And then some people say, I just drink a cup of water and I gain weight. Because everybody's metabolism is different. So we cannot make assumption that everybody is going to eat the same food and they will process or digest it in the same fashion. Because our genetics or lifestyle is impacting our metabolism and how we sense the food, how it is digested and utilized in our system. So food science is going to expand in terms of how it impacts society. And we have multiple other projects. These are some titles. Again, we do some research on dry beans, how stone milling is one of the hot topics these days. We also do some projects on vacuum steam pasteurization. But sometimes I bring it up to my students and, I have all these ideas, but I don't know how am I going to do it. And if nobody is going to come and ask you, you have to be proactive. You have to work so hard to explain that your ideas are good. Sometimes it will be denied, and you should try. So this is why I say if opportunity doesn't knock, you have to build a door and make sure that somebody is going to knock. Gut microbiota is again a hot topic. As I mentioned, that I have a widely developed, very well developed research network, not only supported by university but industry, again, national and international level. So this will be my collaboration and support. And again, anything we do, as I showed you before, I had multiple researchers being part of it. Teamwork is very important. and see, here is the teamwork, but if somebody leaves, it could be team war. And this is not how we spell the war, but it sounds like we will have distraction in the function. So teamwork, working together is very important, and everything we do, success comes from the teamwork. um, it's winter here now, we have snow, but this will be North Dakota State University campus during some spring or summer or early fall, and we have the band. I know I told you so many different topics, different ideas, because I wanted to present you my vision and how we look at different problems from different angles. And and I have given so many presentations, and nobody remembers a number on the slide. Nobody, sometimes they may not even remember the details, but usually they remember how you made them feel. So I hope I made you feel good and you will remember some of the topics that I mentioned. This I have a research website and some social media account. So I will be more than happy to respond any questions you have now. Thank you.
Dear Professor, thank you very much. That was a remarkable and impressive. Thank you very much for all this information. That's for sure enlightening. So Mrs. Kaya wants to give a little speech about the station. Thank you very much again. Mrs.
Okay,
Kaya?
sure.
Yes,
Great. Thank you.
thank you, Teacher Umut. And hi, dear Professor Şenay Şimşek. And I'm very, very pleased to you to come here, and you enlightened us with your words, with your knowledge also. You share all of your knowledge with us. And I'm sure that my teacher, when we listen to you and we feel ourselves get closer to science, and you show us there are a big picture beyond the food chemistry. We cannot see, but you draw to us in our mind also the picture of the chemistry in our daily life also. And I'm so impressed, my teacher, so impressed, and because my regional area also chemistry, and this food chemistry also impressed me, dear teacher. And there are different questions I want to ask you, my teachers. We collect from our students also. And does heating in the microwave disrupt the carbohydrates also?
Oh, so the question is, does microwave heating distract the carbohydrates?
Yes.
Um, so distraction is, I don't like, so this is a question we have to define some of the terms. Good question. That's why I said distraction means that if it is causing hydrolysis, like making it smaller or less digestible or more, no, it's not going to make, but the microwave is going to impact how carbohydrates are interacting with the water in the food sample. How I would say that is most of the times we use carbohydrates because in the foods because they are controlling the water activity. So water activity is very important, especially for food safety. The best example is honey. So honey is very sweet, and if you leave it in your bench for days, it's not going to spoil, right? Because it can flow, but it is not going to spoil. Why? Because the water activity is very low. Every single water molecule has interaction with sugar, so it's not going to the microorganisms are not going to grow in the honey. And think about your bread. If you leave your bread on the shelf, it's going to get some fungi growth 2 or 3 days later. Why? Because, and it's kind of surprising, because bread is dry, something is flowing and it's not getting spoiled. Why? Because of the water activity. So and for microwave, it is not going to change chemistry of carbohydrates, but it's going to change how carbohydrates are having interactions with water. So it, of course, it can impact the texture of the food product because of interactions with water, but for carbohydrate itself, no.
Okay, this is so important for us because we use so many times in our daily life, microwave also. And another question, my dear professor, has wheat also evolved? Can you compare the first wheat and the current wheat?
So the first part of the question is wheat.
Is wheat also evolved? And
Evolved. Evolved. Okay.
evolved. Yeah.
Yeah. So again, this is a relative terminology. So if we go back thousands of years ago, the ancestors of wheats are einkorn, emmer. Even the wheat that we are using today, durum wheat for pasta production, they have there are some changes because of the genome. So today's we call that common wheat. The common wheat is the bread wheat, and it has 6 genomes: A, B, B, D, D. But the ancestors of wheat, they sometimes have diploid or tetraploid. So durum wheat is much older than the durum wheat that you are using to make pasta, is much older than bread wheat. And if of course the genome change, chemistry change, but they are relatives. If the question is thousands of years ago, yes, there is they evolved. But if the question is comparing wheat, bread wheat grown a couple hundred years ago versus today's wheat, we call that modern wheat versus the historical varieties. And we have done studies on that actually, and we found that, and the idea was, someone raised the concern that old wheat varieties, they were not causing celiac disease. You know that as a celiac, right? The celiac disease. And they claim that if 100 years ago people were eating wheat, they didn't have the celiac disease. But today's wheat has lots of these toxic proteins, and we are showing reactions. So we extracted the celiac disease causing proteins, and in today's wheat varieties and the historical ones, and we realized using the proteomics approach, so celiac disease causing proteins were always there. So in terms of chemistry, they are not different, and we did not see significant difference old versus new varieties. And of course there are some changes in the total protein and yield, but modern wheat is not toxic. Modern wheat does not have poisonous compounds. You know, I know some scientists say that if you eat bread, you will have depression, you will have health problems. No, bread is not bad. I am against that claims, and there is no scientific basis for those claims. But if you eat 5 breads a day, of course it's bad. So if you eat at normal levels, it's good. So any food is the same. If you eat too much sugar, if you eat too much fat, too much proteins, so anything you eat so much, of course it's not good. But if you eat at right amount, all these are good. And the modern wheat has changed, but in a good way actually.
Okay, as a result, we love bread, we love wheat, our teacher. Okay, we
Yes.
love, so love. Okay, also there is a big, there should be an area we can talk about your educational technology, because at the starting point, your presentation, we see your educational technology also, and we so impressed about this, because you can touch the molecules visual, as a visual. And
Yes.
in laboratory, we should touch real life, for any component or for any molecules. But you now touch molecules as a visual. And please define us this emotion also. We can, I don't know also the emotion of touching the molecules as a visual.
Yeah, so you have those glasses with sensors. I mean, sometimes even though you try to touch it, it's not there. So it just gives the impression it's there. So yeah, I mean, I am just using the technology for chemistry, but it's the, I'm sure you have heard that it's augmented reality. So now it is finding applications in education, and especially today's world, we cannot put them in the lab. Or I don't have it with me right now, but I was using some molecular models. This is carbon and it has to make 4 bonds, for example. This will be a double bond or single bond, and everybody was passing around the molecule. We cannot do this anymore. You know, we are not supposed to touch at the same time. Those models are good.
Okay. Do you choose this artificial technologies, or you still choose to our old experiment techniques, my teacher?
Yeah, if these are good with the conditions we have right now, it is the best that we can do, because again, we cannot take them into the lab. But if everything is back to normal, I still believe that hands-on, doing together, helping each other in the lab, it's going to be a better experience. Since we cannot do that these days, so this is the best learning experience that they can have, and it will be as close as possible to real world. But I think once everything is back to normal, we should still do the hands-on experiments, and we can still use this technology to supplement. But today, again, we cannot have them in the lab.
Okay, my teacher, there is another question. Can the controlled carbohydrate release systems be formed by using nanoparticles or particles?
Yeah, actually, this is a very hot topic in our field, that there are lots of carbohydrates that including starch, seaweed, polysaccharides, alginate, carrageenan, and they are used to have a slow or controlled release of compounds in food systems or in the digestive tract. And how we can visualize or imagine this is like when you take pills for painkiller or antibiotic, did you realize, and sometimes it's like a candy, you have to bite it with, and um, so it starts to absorb by the body starting from mouth. But sometimes if you take some medicine, if you notice, they are inside a capsule, right? Like a plastic crunchy capsule. Why we have different forms of medicine? Because we want that medicine to melt in different parts of our body. And when you take those capsule types of medicines, like I think I had in, maybe not in this presentation, but so those, we don't want that digestion to start in the mouth. We want it to start in stomach and slowly move into the small intestine. So that is why they are inside the capsule. Same thing. So you can actually prepare some of those, especially in the gel form, some compounds, they could be forming complexes and carbohydrates, especially again the ones that I listed now, alginate, carrageenan, or starch. They could be utilized as a slow releasing
Okay. My teacher, you said us also wheat is not poisonous, but there are two
that's what I believe, and I know there are other opinions. Yes.
Yes, but there are two questions more I want to ask you. Sugar, salt, and flour are considered white poison. Is this true? And do carbohydrates have a narcotic or addictive effect?
Yeah. So sugar, salt,
And flour are
flour.
considered white poisons.
I
This
yeah
is announced so many times in
yes,
our.
yes. No, I respect. So, has anyone taken? Did you take a food toxicology course? I think the previous speaker was a medical student, right? Was he in the medical field? Coronavirus prevented?
He also is a student in the Middle East Technical University.
No, no. Does anyone?
dear professor, he's a
Yes.
chemistry student, not
Oh,
the
chemistry. Okay.
medical area.
Okay. Just
So
because
that's
I
why
thought,
that's
yeah.
why our young scientists ask you what can they do,
Yes,
because you know
I
very
was going to say that there is a very well defined slogan in any toxicology class. If anyone has taken a toxicology class, we always say poison is in the dose. Poison is in the dose. So if you eat too much of something, it could get poisons. But salt, for example, it's not poisons. People have been using sodium chloride to enhance flavor or mask some flavors in the food for many years. But if you eat too much, then it can cause problems. So the poison is in the dose. And again, for sugar, sugar is good, but if for so when I drink my coffee, I just put little bit sugar, and it's not poisons. But if you are eating too much sweet, then of course it's going to impact your blood sugar levels. It can induce insulin production. And again, for flour, it's not poisons. People have been eating bread for thousands of years. Who passed away because of eating bread? It's not poisons. As I told you before, if you keep eating bread too much out of portions for a long time, then you will get obese, and you might have diet-related diseases. So I don't believe that those will be poisonous if you consume them in the recommended levels. So I don't believe in that. Again, just who can eat a cake without sweet taste? Who is going to eat, any food with no salt in it? So you can decrease them, use them little bit, and it's okay. Poison is in the dose. It's the main message of the toxicology class. That's why I ask. Uh, I didn't want to direct the question to him, but this is again, if anyone has taken a toxicology, they always say poison is in the dose. And the second part of the question, what was it?
This is the do carbohydrates have a narcotic or addictive effect?
No, no. It's just a habit. If whenever you eat something salty, you are craving. Those are cravings, but addictive is again. That is why I called him if he was the medical doctor, because addictive means it's a chemical process, um, like having addiction to smoking or alcohol. It's even crazy idea comparing food as addictive as compounds. It's the habit. um, if you are let's say taking a walk every morning, or you have routines, you might have that, oh, I'm supposed to do this after that, but there is no chemical or hormonal reasons that you will be addictive to carbohydrates. Again, I know there are claims out there. I respectfully disagree.
Okay, thank you. There are another question. This is maybe the last question. Mrs. Şimşek, have you any advices for a freshman year chemistry student who is interested with food science?
Yeah, so chemistry is a great undergrad degree for me. I think it's good because it's one of the basic sciences, and we understand how molecules are interacting with each other. We learn all the analytical tools. And then when you are in an applied field, so food science is basically an applied chemistry. Or, I strongly suggest, so anyone, actually, if someone is having the chemistry degree, you can build on your career on maybe medicinal chemistry, environmental chemistry, some engineering aspects, like I did in the food chemistry or biochemistry. So there is so much that one can do. And do I recommend it to a freshman chemistry? Sure, if you have passion for it. So when I was having my degree, PhD in food chemistry, I really liked the idea to seeing all my chemistry information was basically coming into life with food chemistry. Like for example, like again the starch chemistry, or that water molecules having interactions with starch, and it turns into a product you can eat. So that's why I like it. It has the basic component, but it's applied and it is so relevant. Everybody eats. You, if you are a human, you have to eat. So we do a science on an area that is related. It's not, you cannot skip eating. So that's why I like this field. If anyone has passion, I strongly recommend.
Okay. Can you take a fruit supplement to reduce its harmful effect with foods containing carbohydrates to avoid diabetes?
So is the question is, can we use some supplements to prevent diabetic effects of carbohydrates? Is that
Yes,
what?
teacher.
Um, I would refrain providing any response for this, because it depends. If you are a healthy individual, um, we always suggest that all the nutrients should be taken by natural ways, eating veggies, fruits, right amount of food. And also timing when you eat is extremely important. I failed to mention that. I forgot to mention that when you eat is extremely important. If someone says, "Oh, I don't eat anything," but just before they go to bed they eat, then it's not good, because your metabolism is slow. Then you can have undesirable effects. So supplement I wouldn't recommend. I think this question is about, there are some supplements, they inhibit the enzymes in the digestive system. Maybe it's targeting that. We never recommended those. Just poison is in the dose. If you are eating too much, you will gain weight. If you are eating too much sugar, it's going to impact your insulin levels and diabetes. Salt is high blood pressure and other related disease. You don't have to completely ignore them, just use them in the right way.
Okay, thank you, dear teacher, again. And does carbohydrate consumption have an effect on mental clarity?
No. This is like, just no. There is nothing. I am not going to defend it. Again, there are claims out there about it, but no, that
Thank you.
I don't think there is any scientific basis. All those are those claims. I have read them, and yeah, they are not biased studies.
Okay, thank you. And which foods should we consume to be efficient in school and business life?
Yeah, actually, I am not a dietitian, but so this will be a dietitian related questions. But we work with USDA, United States Departments of Agriculture, and they have the like plate picture, share my plate, and they always recommend at least 50% of grains being whole wheat, like whole wheat products. In Turkish, it's like tam buğday ekmeği, right? Whole wheat is good. They also suggest certain portions of vegetables, fruits in our diet. But again, I suggest the time of food that we consume and how much we eat, when do we eat, and um, and again, we have to have the right balance. Okay, balance is the key word. You it's not going to hurt you if you eat a piece of cake, maybe for lunch or for 5 p.m. But if you are eating it late at night, you do not have physical activity to burn it, so it will be deposited. um yeah, so I would say that there are dietary guidelines, and it's balance of fruits, veggies, proteins, and whole grains. Those will be what we recommend. And in the US, also in school systems, they follow those guidelines. This is how food is offered to children.
Thank you, dear professor. And I'm also so thankful to you because you get earlier for us. You give.
Oh, yeah, no, I get up early actually, so it wasn't that bad. Just maybe 1 hour early, but it's good. I'm happy to be here with you today. Thank you.
Really, we are happy be with you, and you share your words and your knowledge also to us. And I feel more better after listening to your lecture also, my teacher. Thank you so much. Thank you. Greetings from Turkey. Greetings from your country also. And please, Umut teacher.
Yeah, thank you, Filiz, again for inviting me for this awesome event. I did my best, and at the end of my presentation, I hid my website and social media. If anyone wants to follow up, please do so. Or Filiz has all the information of me in terms of phone numbers, emails. So I will be more than happy to get in touch later as well. Thank you.
Thank you.
We thank you. You are a great scientist and you are representing our country in USA, and we are thanked to you, dear professor. Thank you very much. And it's a great pleasure to have you today. Thank you very much again.
Well, thank you. It's very humbling to hear this from you. Thank you.
Thank you very much. Okay.
Thank you.
So it is the end of our meeting, but before you leave, dear students, we got a polls to fill up because we have to improve our INTOC lessons. So if now a post will appear in your screen via Zoom, so please fill up this poll and tell us how the INTOC improve himself. We are waiting. Mr. Çelik, you can start the poll. Thank you very much.
This is great. I haven't seen this before, so I learned something here. It popped up in my screen, but I didn't fill the form. So
because you are professor, you are the host of this meeting. You are the owner of this meeting. You can do that.
Yeah, I know I didn't, but it's very good. So I haven't seen this before. It's very good.
After 6 months online lesson, you can learn a lot of things from Zoom.
Yeah, this is good. So should I exit this meeting or?
Sure, my teacher. Sure, my
Sure.
Thank you.
Thank you very much attending
might
again.
More lessons maybe, but yes, I will sign out again. Thank you so much, and have a great night. Have a great weekend, everyone. Nice to meet you, everybody. Bye.
Bye. Thank you.
See you later, teacher.
Thank you.
Is there any problem about sharing?
No, 20 of the 51 students are filling our polls right now. So everyone is here right now. Please, please, please. We are waiting to attend this post because it's very important for us to improve ourselves. Also, we have to learn. We want to learn your opinion about us.
Hocam Çelik Bey duyuyor mu acaba?
Thank you very much for attending this post. Teşekkürler. So, in the last verse of today's meeting, that was quite remarkable. We had three different and three remarkable scientists for today. For information, the INTOC will continue next month with the career science. So please prepare your subjects and join us next month. Thank you very much for everyone to joining us. We can't wait the other meeting for the next month. We see you in the March. See you later.
See you later, our dear participants. See you later.